I'm Dr. Laura Kilpatrick. I'm a postdoctoral researcher at the Centre of Membrane Proteins and Receptors at the University of Nottingham, and I'm currently investigating and quantifying the pharmacology of the vascular endothelial growth factor receptor, VEGFR2, and its co-receptor neuropilin-1, and these are both receptors that are heavily involved in cancer progression, and so they're very attractive anti-cancer therapeutic targets. In respect to what we do, the vascular endothelial growth factor receptor, so VEGFR2, there's not really a lot of known quantitative pharmacological data for it, particularly in isolation where it's not expressed amongst other VEGF binding proteins. So we've been able to do a lot of model cell system work so we can try and isolate the pharmacology of that receptor.
We're looking at the VEGFR2 receptor, which is already targeted by a number of different anti-cancer therapeutics. However, they largely lack long-term efficacy. Therefore, we want to find new ways in order to target this receptor. So in particular, we're looking at ligand binding. For this, we use fluorescent ligands and looking at the bioluminescence resonance energy transfer. By looking at how existing drugs target this binding interaction, we can develop new therapeutics that target this more specifically, so it has fewer off-target side effects, it's longer lasting, it doesn't have these resistance mechanisms. We use the PHERAstar in order to look at the real-time binding kinetics, so looking at how these different forms of VEGF bind at VEGFR2 in real time at 37 degrees, so it's got this physiological temperature and we can see how it interacts. This temperature control is particularly useful for looking at kinetics, because kinetics can be widely varied by temperature.
We know how important kinetics are for ligand binding and long-term residence times for drugs, so we can really begin to sort of tease out that using the PHERAstar. The great thing about it for what we do is you can not only measure fluorescence but luminescence as well, and the even better thing for us is we do a lot of bioluminescence resonance energy transfer assays, and that requires the simultaneous measurement of luminescence and fluorescence emissions, and the PHERAstar lets us do that. And it's also really easy to use, and the great thing is, you know, I can put a plate on and I can get readout within 2 minutes, which is fantastic.
My research looks at a GPCR chemokine receptor called CXCR4. I'm looking at how that interacts with other proteins within the cell. What we really want to work out is how CXCR4 is working in the endogenous environment, so in a physiological or pathophysiological condition. With my gene editing techniques I've been using, looking at really low endogenous levels of receptor expression and very low signals, the PHERAstar data we're getting is giving us a better idea of how this receptor is working in a disease state or in normal physiology. One of the really good things about the CLARIOstar that we've been able to develop is that we can look at a multiplexed assay, so look at how CXCR4 interacts with more than one protein simultaneously. When we're investigating CXCR4, we normally use model cell systems that are using overexpressed receptors. So one of the really cool things that we're able to do with the PHERAstar, as well as the CRISPR-Cas9 genome editing technology, is look at CXCR4 in a normal physiological environment or a pathophysiological environment. So this is really allowing us to unlock the function of the normal CXCR4, pathological CXCR4, in a disease state.
A lot of the work I do currently is using model cell systems, which is great because you can control what you're looking at, but obviously that doesn't necessarily reflect physiological conditions. So I think now with the advent of gene editing techniques like CRISPR and some more advanced luminescence and fluorescence-based techniques, we can begin to apply this quantitative pharmacology in more physiologically relevant cell types, which I think is incredibly important for future drug discovery efforts.